Great, let's get started. Welcome everybody to Hutch at Home. I'm Jeannie Chowning. I'm the Senior Director of Science Education at Fred Hutchinson Cancer Research Center, and we are delighted that you are here with us today. As you know, this is an ongoing series, happens every Tuesday from 4 to 5, so please put us on your calendar. It's always the same Zoom link. Really excited today to hear about playing hide and seek with cancer from Miranda Lahman. And Miranda is a graduate student in the Chapuis Lab at Fred Hutch, and I'm really looking forward to her presentation. Just a quick reminder that this is a talk geared towards science teachers and students, and so if you go to a different group, please prioritize the questions of the science teachers and students. Please mute yourself if you're not talking via a phone. Towards the end, we'll open it up to questions and you can feel free to unmute yourself. And feel free to add questions into the chat as we go as well. And when there's a pause in the presentation, we can address those. And lastly, just take a moment to complete our feedback form at the end of the talk if you are so inclined. It's always good for us to hear from who have joined us. So without further ado, I'm going to turn the screen over to Miranda.
Wonderful, thank you. So let me share my screen. Looks like people can see it. So I'm going to talk to you. Thank you all for coming to the Hutch at Home today. As Jeanine said, I'm Miranda. I'm a graduate student in the laboratory of Dr. O. Chapuis, who focuses on treating acute myeloid leukemia with immunotherapies. So I'm going to talk to you today about a little bit about my research and how we play hide and seek with cancer. So in this talk, I'm going to give you a little bit of an overview. First about me and how I got to where I am today as a graduate student here at the Hutch. I'm then going to talk to you about immunotherapy, give you a little background as to what it is and why we use it in the context particularly of bone marrow transplantation, and talk about the clinical trials that I run out of the Hutch using engineered T cells, which is really the focus of the work of the Chapuis Lab. And then I'm really going to talk to you guys, this is sort of the focus of my research, is what we can learn from failed therapies. So when the therapies don't work, what can we learn from them so that we can better engineer our therapies such that that doesn't happen again in the future. And then finishing on a high note with this bright future of cancer research. So a little bit about me, basically from pinecone enthusiast to research scientist. So many people have very different journeys as to how they come into science and how they decide they want to do research. And for me, from a very young age, I knew that I wanted to do something with science. So even way back as a kindergartner here when I was student of the week, I said that I wanted to be a school nurse when I grew up. And this was on top of that initial love of medicine. My interest in science really was fostered by my family. So my mom and dad would take us outdoors. We'd do a lot of camping, and I would love to investigate the surroundings. So I would pick up pine cones and investigate the moss and the animals. And so it was always something that I was really interested in was biology. It's kind of funny, I used to ski and I would stop and pick up pine cones on my way down. And so my dad didn't like skiing with me at the beginning because I was too slow. I spent too much time picking up the pine cones. And then my grandpa was also pretty influential here. He was a medic in World War II and continued to practice after coming home. And I was always really fascinated by medicine from his perspective and understanding sort of how medicine had evolved from sort of more earlier times to now. And I really sort of became very interested in it. And I always loved it in school. I always loved the aspect of it that you really got to know sort of what was happening in the world. And this really was exaggerated when I got to high school, so I had a fabulous AP biology teacher, Mr. Title pictured here, and I fell in love with the molecular aspect of biology. So I really loved learning about the smaller and smaller sides of biology. So I did like big picture biology, evolution or geology, but I was captivated by the small inner workings of the cell. So after that, I moved from Colorado out here to Washington to attend Western Washington University, where I joined my first research lab as a junior. And in that lab, I actually studied plants, and we studied how these plants could be infected by a certain fungus called Aspergillus. And that's really important in the context of corn, because this fungus infects corn, which is a major food crop. And as you can see here, we were a very serious lab, and we had no fun at all. You can see me here over in the corner as the infecting fungus, and my lab mates, as well as my lab professor, are the Arabidopsis plants that I can infect. After that, I graduated and moved down here to Seattle, where I worked at a small biotech startup called Stratos Genomics, where they worked on a DNA sequencing platform. And from my experience there, I found out that I really wanted to do research, and I really wanted to pursue a career in research science. And at first I thought I really wanted to pursue researching HIV. And so because I work so much with sort of the small sides of cells, I work with individual cells, individual components of cells, I wanted to get a more humanistic aspect to my research. So I went to an area of the world where HIV is really prevalent, down in Zimbabwe. And then it was here that I realized I really don't want to do infectious disease research. Even though I was fully aware of how HIV was transmitted and knew that it was impossible to get it from just laboratory testing, I found myself very anxious all the time, even though I knew that you couldn't spread HIV by shaking someone's hand or any of those things, or it couldn't spread very easily on surfaces. I still found myself very anxious. So I pivoted, and I ended up deciding to instead study cancer at the University of Washington. I'm part of the M3D program, which stands for Molecular Medicine and Mechanisms of Disease. And it's a wonderful program, and I get to carry out my research in cancer here at the Hutch. Now, outside of research, I think it's important to know that research scientists still have a lot of fun outside of the lab. So I love to run. I ran competitively both in high school and in college. I love to ski. I love to be in the mountains, whether it's hiking in the snow or hiking in the sun. I just love to be in the mountains. I also love to travel. I've traveled all over the world, including Morocco, Iceland, and the Netherlands. I also like to mountain bike, and I'm an animal lover. You can see me here with my cat, Mina, and my dog, Nala. Okay, now for the research. So my research, as well as the research in the clinical research division at Hutch, really focuses on translational research. And what that does is it takes findings and understandings that we learn from the lab and uses those to generate therapies that we can translate to the clinic. This translation is often a clinical trial. We then take the lessons learned and information that we learned from the clinical trial and use that to go back to the lab where we engineer better therapeutics to improve treatment for these patients. And this sort of cycle is called the bench to bedside cycle. And my research really focuses on this, coming from the clinic back to the bench so that we can go from the bench back to the clinic. Okay, so our immune system fights off harmful pathogens. For those of you who don't think about the immune system on a daily basis, this is a little bit of a review. So there are a number of different players in our immune system, but they really focus on targeting outside harmful pathogens, whether that be a virus or bacteria, or even cancer. And there are a number of different players. There are many more than the 4 that I have listed here, but I wanted to draw your attention to these guys. So we have a CD8 T cell, or called a killer T cell. And this CD8 T cell really finds and kills infected cells. Then you have something called a B cell. And a B cell is what produces antibodies. I'm sure you guys have all heard of antibodies. Well, they're made by B cells, part of your immune system. You also have something called regulatory T cells. Now, these regulatory T cells keep some of the other T cells in check, and they're really important in a number of different diseases, including autoimmune diseases. I'm not going to talk about them much today, but they do play an important role. We also have something called a CD4 T cell. They're often called helper T cells. Now, these T cells sort of help the cytotoxic or the CD8 T cells find and function so that they can effectively find and identify their targets. I'm really just going to focus on these two guys for the remainder of the talk. As I said, you've got your cytotoxic T cells, which are killer T cells. And I think this comic really captures the essence of a CD8 T cells. He's ready to find and kill those infected cells. Oftentimes that's referring to something like a viral infection, but it can also refer to something that's not per se infected, but just something that's not right. So maybe a cancer cell. And then you have B cells that can make and shoot antibodies to tag something like a virus or bacteria that indicates that that molecule is bad and tags it for degradation. Now, killer T cells can really target not only infected cells, but cancer cells. So one of the main components of cancer, it's a very complicated disease. So don't be overwhelmed by this figure. So what this figure is really trying to tell you is that there are all these different hallmarks of a cancer. So it grows really rapidly. It evades the immune system. So that's a characteristic of cancer, is that it can actually hide really well from our immune system. So as scientists, we want to be able to take our immune system and revamp it and so that it can see and identify this cancer. There are a number of different other hallmarks of this disease that I'm not going to go into, but are all areas of active research at the Hutch and around the world. So immunotherapy, what is it and why do we use it? Immunotherapy is really defined as a substance that modulates the immune system. Now, if you think of the immune system per se like a car, an immunosuppressor would be something that puts the brakes on the car. So it really stops that immune system from overreacting. And immunosuppressors are often used in the cases of perhaps autoimmune diseases where your immune system is attacking your own body. Or perhaps in this scenario of organ transplants or bone marrow transplants where you need to suppress the host immune system so that the transplant organ can survive. Then you can have something called immune enhancers. Now, immune enhancers are like taking the brakes off the car and allowing the immune system to accelerate. Now, this is what you really want to do in cancer scenarios where the cancer is hiding from the immune system, and you want to take the brakes off the immune system so that it can see, identify, and get rid of the cancer. So in the context of bone marrow transplant, that's almost what you're doing, but instead of taking the brakes off your own immune system, you're giving the patient a new immune system. So bone marrow transplant is a form of immunotherapy. And it starts with a recipient who is probably receiving some sort of anti-cancer drug, and they receive some radiation. And what those do is they actually sort of ablate the recipient's natural immune system. And then they take bone marrow from a healthy donor. Now, many of your immune cells actually originate in the bone marrow. So this is where they sort of start, sort of the birthplace of many of the immune cells. So by transplanting the bone marrow, you're able to give the recipient new bone marrow and effectively a new immune system. And that immune system can hopefully identify and see the cancer, specifically the leukemia in the bone marrow, and eliminate it. Now, this is often done in the context of acute myeloid leukemia, which is the disease that I study. So AML is a really heterogeneous disease. It has all these different flavors of leukemia. And so its survival actually lags substantially behind that of other blood cancers. On top of that, it's one of the most common cancers in the US and most common acute leukemias in adults. So at 20,000 cases per year, it accounts for about 80% of the cases. And unfortunately, AML is not cured by chemotherapy. But there is a bit of a silver lining in that stem cell transplants, and so stem cells or bone marrow transplants, those are names for the same thing, does cure some AML. And we know that that curative effect comes from those T cells in that bone marrow responding to the recipient's AML. So we know that the T cells from the bone marrow transplant are really important in this curative effect. So then as scientists, it should come as no surprise that we wanted to engineer these T cells. And so we wanted to take those T cells that we knew were really important and engineer them so that they could specifically see and eliminate the leukemia. Now, these engineered T cells have actually shown quite a bit of clinical promise. Okay, so these T cells, what are they doing exactly? So they see peptides presented on the surface of the cell. And so what does that mean? So all cells, including cancer cells, communicate to the immune system through these presentation molecules. So what a cell will do is it will take proteins from the inside of the cell and it will chop them up with something called the proteasome. Now the proteasome sort of acts like a molecular scissors and chops up these proteins into little pieces called peptides. So peptides are really just small pieces of proteins. Now these peptides can be various sizes, so various lengths of amino acids. So amino acids are the building blocks of protein, and they can be, say, 9 amino acids in length, they can be 14 amino acids in length, they could be 2 amino acids in length. But the ones that are about 8 to 14 amino acids in length get presented on these presentation molecules called HLAs. Now these HLAs come in a variety of different flavors and present these peptides to the immune system. What's really interesting and important about these HLAs, they stand for human leukocyte antigen, is that they are what you need to match, part of what you need to match in an organ transplant. So for a bone marrow transplant or an organ transplant, you'll hear of having a matched donor or a partially matched donor. And so one of the things that plays into that matching is these HLAs. So not everyone has the same flavor of HLA. And the immune system can actually attack those if they don't match enough. So the T cell interacts with this HLA peptide complex. And this is a very specific sort of lock and key interaction. And this TCR HLA peptide interaction, if it's strong enough, will actually signal the T cell. And it will signal the T cell that, hey, this isn't me, this isn't right. This is maybe a virus, or maybe this cell just needs to go. And so it will tell the T cell to produce these things called perforin and granzymes. And what those do is they poke holes in the membrane of the cell, causing the cell to explode and lyse. The T cells will then go on their way and repeat the process as they interact and encounter the correct peptide epitope or peptide HLA presentation complex. Now, there are a number of ways that we have decided to engineer T cells. Probably the one most people know of is a CAR T cell. Now, CAR stands for chimeric antigen receptor. Now, chimera means sort of a mosaic, or like more than one piece of something. And so that chimeric part comes from this T cell actually doesn't have a T cell receptor on it. It has sort of the head of an antibody. So these antibodies, remember those B cells make those. So this CAR T sort of has the head of an antibody and the tail of a T cell receptor. So it can recognize an antigen and signal to the T cell like a regular T cell receptor would, and tell the T cell if the antibody encounters something to go ahead and produce those perforin and granzymes to poke holes in those cancer cells. Now, what my lab does is something a little bit different. We've engineered something called a TCR T cell. And instead of putting an antibody on the outside of that T cell, we've put a very specific T cell receptor. So T cell receptors come in more varieties than are easy to test. And they're very, very specific. But what you can do is you can kind of fish them out of pools and identify the T cell receptors that are really good at seeing and finding cancer. That's something that my lab has done, and we've pulled out some of these T cell receptors that can really see and target cancer. And we used those in the clinical trial. Now, what's important to note is that there's a big difference between CAR T cells and TCR T cells. Now, CAR T cells can only see proteins that are on the surface of the cell. So they're very limited in how the types of proteins that they can see. Now, unfortunately for AML and a lot of these myeloid malignancies, the surface markers on these cells are shared with really critical stem cell populations and really critical cell populations. So CAR T cells are really good at targeting sort of B cell malignancies and things that you can live without a B cell actually. But in myeloid malignancies or AML, they share surface markers with really important cell types like red blood cells or give rise to red blood cells. And so you can live without an antibody, but you can't live without red blood cells. So it's really difficult to target these with CAR T cells. So we target them with TCR T cells so we can really look at all of and target all of the proteins inside the cancer cell. Now again, this TCR-HLA peptide interaction is at the heart of TCR T cell therapy. Like I said, we've picked out these T cell receptors that are really good at seeing and recognizing cancer, and we put those into a T cell. Now to generate these products, we take blood from a patient. That's called a leukopheresis. So we take blood from a patient, and the patient's blood has all these different types of immune cells. And we pull out just the CD8 T cells. We then stimulate them to expand them to get them to divide and make lots of CD8 T cells. And once we have lots of CD8 T cells, we can actually give them our T cell receptor of choice. Once we give them this T cell receptor of choice, we can expand them yet again so that we have even more T cells that we can then put back into the patient. So basically what we do is we take patients' own immune cells, own blood, we soup them up so that they can see the cancer and put them back into the patient. And this is exactly what we did here in a clinical trial led by my boss, Chapuis, as well as a leader in the field, Phil Greenberg. And so what they did is they took high-risk AML patients post-bone marrow transplant. And then they gave these patients transgenic T cells. So those T cells that were given that TCR to see and recognize and eliminate that cancer. These patients were split into two groups, and they either received these cancer-fighting T cells before they had detectable cancer relapse. This is called a prophylactic arm. So prophylactic means sort of before, so the treatment is before the disease. Now, the other part of these patients received these cancer-fighting T cells after they already had detectable AML. So in this arm, they wanted to prevent AML from coming back, and in this arm, they wanted to eliminate the already existing AML. Now this arm, the prophylactic arm or the preventing AML arm, actually showed a lot of success. And don't be too overwhelmed by these graphs. The left side here are these things called Kaplan Meier plots. And so how a Kaplan Meier plot works is you have the percent people over here, and each time a patient or a subject or a subject in the study passes from their disease, the line goes down. And so if you see the line decrease, that means people are succumbing to their disease. And then if you don't see the line decrease, it means that all of these patients have survived survived without any relapse of the cancer. So the cancer doesn't seem to be reoccurring, compared to matched controls who really have seen quite a bit of attrition or reduction in size of the number of people there, not the size of the people, the number of people there. So about 50% of those who did not receive the T cells actually succumbed to their disease and passed away. These two lines denote either overall survival or disease-free survival. So overall survival just means if you're still alive, you count as being as overall survival. And this disease-free survival, you can actually live at quite a while with this disease, either at a low level, and maybe it's a little less aggressive. And so if you have no evidence of disease, it's considered disease-free survival. So as you can see, this clinical trial was quite successful, and these T cells were able to prevent the relapse of the AML, which we see in these graphs over here, where all 12 patients enrolled in this arm of the trial did not experience any relapse, compared to about 30% of those in matched controls who actually did experience relapse. So this is great. They have a lot of clinical promise. But when we used these cancer-fighting T cells to see and identify existing AML, they didn't perform quite as well. So again, we have this Kaplan Meier plot where we see the matched controls being in this black dotted line, and the red line is our treatment arm. So we had a number of patients, unfortunately, pass and succumb to their disease, and so we didn't really see a significant improvement. So we wanted to know why these T cells were working so well when they were given before detectable AML, but were unable to eliminate existing AML. And so this is really where we see this bench to bedside conversion. So we made T cells that we knew were important and engineered them in the lab, gave them to the patients, saw that the patients weren't responding, so we needed to go back to the lab to try to figure out what's going on so that we can change our T cells so that it doesn't happen again. And that's exactly what we did. So again, I want to remind you that it's this interaction that's really important into making these T cells work. So we wanted to know why these T cells were no longer to really engage in this interaction. But first, we need to know what makes a good target for TCR-T. Well, really effective TCR-T cell therapy relies on picking a good tumor-associated protein off the get-go. So a good tumor-associated protein is only expressed in cancer cells, is expressed at a really high level, and ideally it should be required for the tumor to grow, such that if it loses that particular protein, the tumor doesn't grow very well. And then most importantly, we don't want there to be high expression levels or expression of this target protein in healthy tissue. Otherwise, the T cells will see and hurt that healthy tissue. Now, what we've targeted in this clinical trial is something called Wilms tumor antigen 1, or WT1, and it does meet this criteria. It's overexpressed in AML and has almost no expression in healthy tissue. But in addition to that, you actually have to get the peptide from the protein presented on the surface of the cell so that the T cell can recognize and identify that cancer cell, which means that that peptide or epitope has to be processed and loaded onto that HLA. And in addition to that, the HLA actually has to be there, right? I said that there were a lot of different flavors of this HLA. So the HLA that you're targeting with this T cell receptor has to be present in this patient. We've targeted something called HLA-A2, which is very prominent in the Caucasian population and here among people here in the US. So it's a fairly good target for the United States that shares that predominant phenotype. However, it's not necessarily a good target for other areas of the world. For people in Asia, this is not a super common epitope. For people in South America or Africa, again, not a good epitope, or sorry, good HLA to target. So that is something to consider when engineering these T cells is you want to pick, You want to be able to make a number of these T cells that can work with a number of different patients depending on their HLA type. And that's something that the field is working towards, is generating these T cells for more than just a few HLA types. But you have to start somewhere, and so we started with HLA-A2. So we targeted a particular part of the WT1, amino acid residues 126 to 134, that's affectionately known as RIMFNAPL. So each amino acid has a one letter representation of it. So this represents the amino acid sequence. And sometimes things in science are named very scientifically, and sometimes we just sound the word out. So we call this epitope RIMFNAPL. Now, as I was saying earlier, if you need to get this peptide onto the surface of the cell, which requires chopping it up with the proteasome into that peptide, loading it onto the HLA. So if you disrupt any part of this processing and presentation can actually lead to tumor escape. So you could say, lose your WT1. If you don't have WT1 expression, the T cell can no longer recognize the tumor. So the cancer is able to hide from our therapy. You can lose the HLA. So we have a lot of these HLAs. The cell doesn't need all of them. So it can selectively hide the one that the T cell is targeting, sort of making these tumor cells bald. And again, our T cell can't see the tumor. And then lastly, what you can do is you can change the way that the proteasome chops up these proteins into peptides, such that you no longer get the peptide that's presented on the HLA. And again, the T cell can't see the cancer. So cancer is really sneaky, and it's actually really good at manipulating itself and morphing so that it can change the way that it looks so that our immune system can't see it. And this particular example where the proteasome composition changed, we actually observed in one of our patients in that second arm of the clinical trial. We observed this patient actually change the type of proteasome that it expressed to escape our T cell therapy. So this led us to hypothesize, well, why don't we target peptides that are produced regardless of what proteasome type is in the cell, so that we could enhance our T cell therapy? So indeed, turns out you have more than one type of molecular scissors that chop up proteins. You have something called the standard proteasome, and you have something called the immunoproteasome, which are actually very similar, but they differ by only 3 subunits: beta 1 for the standard, 2 and 5 for the standard proteasome, and beta 1I, 2I, and 5I for the immunoproteasome. Now they work very similarly in that they produce, both produce peptides, and they do actually produce some of the same peptides. Those peptides are loaded onto the cell, or loaded into HLAs and presented on the surface of the cell. So we want to target these mutual peptides. So to do that, how might we test if our TCR T cells, so our engineered T cells, can actually see a mutual peptide? Does anyone have any ideas? Okay, it's okay if you don't. So how I test this is I take a cell line and put it in a co-culture system. So I take cell lines expressing either the standard proteasome or the immunoproteasome, and I take them and I put them in a dish. So I take those cells and I put them in a dish, and then I co-culture them with the T cell expressing either, say, TCR1 or TCR2. And then I want to see if those cell lines expressing the immunoproteasome or the standard proteasome still exist. If they're there, that means that my T cell couldn't see them and couldn't eliminate them. If they're gone, it means that my T cell could see them and got rid of them. And I did exactly this. And it turns out that I was able to find a TCR that does see an epitope of WT1, so a different part of WT1 that gets killed by the TCR. So it can kill cells expressing either the standard proteasome or the immunoproteasome. And unfortunately, the TCR T cell that went into clinic, the one that was used in that patient, can actually only see cells expressing the immunoproteasome. So that tells us that we need to pick better targets, and we need to think a little bit more critically about what piece of the protein we target and how it gets on the surface of the cell. And that's something that we and many other people are working on, is to find ways that we can really identify those mutual peptides. And that's what we're doing right now. And so there's really a bright future, because we found this mistake and we're working to get around it. So currently TCR2 is going to go into an upcoming clinical trial. That's very exciting. And we want to use new peptide discovery tools to really identify TCR-T targets, such that we can target those mutual epitopes or mutual peptides and eliminate one of these mechanisms of escape. Now, cancer is really sneaky, like I said, so it might be difficult to do that. However, we're working to eliminate at least one of those ways that the cancer can hide. And teams all over the world are addressing many of these problems, not just the processing of the protein, but how do we get the HLAs to be up there? How do we target more than one protein so that if it loses the protein, we can still target the cancer? So this is a very active and new field of study that people all over the world are trying to figure out. So in summary, I told you about how I got here, and believe me, it's a process. You can enjoy the journey. So took lots of detours on the way, and found myself here in Seattle. Immunotherapy shows a lot of promise, but there's still a lot of work to be done. We still need to figure out how some of these things work and why some people don't respond. And this is one of the biggest things to learn from science, that you can actually learn way more from your failures often than you can from your successes. So we now know that presentation and processing of these proteins is really important in these immunotherapies. We wouldn't have known that otherwise. So it's really important to read and interpret the meaning from your data, and then gather more data and see if it supports your hypothesis, and really engage in that scientific process. And lastly, we really have a bright future for cancer research. There are a lot of people researching this, and most importantly, there's a new generation, the high schoolers and the people teaching the high schoolers, who can think creatively to attack these new problems in not only the field of cancer, but fields all over disciplines. So with that, I'd like to thank everyone in my lab and remind people that science is really a team effort. We can't do this by ourselves. It requires so many different people, people that treat the patients, people who analyze their cells, people who make the cells for the therapy. It requires a lot of people to make these clinical trials work, and then they have chances to be very impactful. With that, any questions?
Thanks so much, Miranda. We'll give folks a minute. Oftentimes people enter questions into the chat. So I invite you now, if you want to enter questions into the chat, to do so. And we'll also, if you'd like to unmute to ask a question, that's great too. I was wondering, could you talk a little bit about how you actually get the T cell receptor into the cells?
Yeah, so the way we get the T cell receptor into the cells is we actually use a virus. So viruses are really good at getting into cells. And so what we do is we package some DNA, some genetic material that encodes for that T cell receptor, into the coat of a virus. So we take the outside of the virus and we put the T cell receptor into the inside of the virus, so the genetic material in there. And then we something called transduce those cells so that they dump the genetic material into the nucleus so that it gets transcribed and expressed on the surface of these T cells.
Good, thank you. There's a follow-up question. Why do you guys use a virus as a way to dump genetic material in? It's kind of a follow-on to my question.
So this technique was developed many years ago and has been optimized for use in the clinic. So we can fine tune how many copies of the TCR get into the cell, because you want to add the genetic material to the cell, but you don't want to add too much, right? Because that's not good for the cell either. So this technique has been fine-tuned. It's also approved by the FDA. There's a fancy new technology called CRISPR that's a bit more specific than a virus, and people are working on using that technology now as well. But for now, the virus is very effective at doing it because viruses have evolved to sort of dump their genetic material into cells and capitalize on the cell machinery to make the virus. So we use part of the virus that's really good at getting material into the cell, but then tell it what material to use. So really just history.
Then a question about the endogenous or the original T cell receptor. Is that still there once you put in the new modified T cell receptor? Is the original one that the cell has still there?
Yeah, it is. And that's two things there. It is still there. And so you might think, well, if the endogenous T cell receptor is there, like it can signal through that one, which is true. And so what we actually do is we take not only CD8s, but we actually take CD8s that can see a something called EBV, so Epstein-Barr virus, which is a very common virus in the population. Pretty much everyone's had it by the time they're maybe 5. And so we take a very common virus, and so what we do is we pull out the CD8s that are specific for that virus. So the endogenous TCR in our product is actually a virus-specific TCR. And part of the thought process behind that is that if the endogenous TCR is there and does signal, it'll be signaling through a virally infected cell. So it won't be targeting something that it shouldn't. It will be targeting something that probably should go away anyway. And on top of that, our T cell that we put in is overexpressed. So it's the dominant T cell receptor on the surface of these cells. Does that answer the question?
There's another, oh, go ahead, Danielle. Was that Danielle? Did that answer your question?
Sorry, I was just saying, yes, thanks very much, thanks.
Okay.
And you were mentioning to me, I think, Miranda, earlier that also there were some moves to use CRISPR to remove some of the endogenous receptors. Am I remembering that right?
Yes, you are. So people are working, our lab included, to actually remove the endogenous T cell receptor. And that CRISPR technology is really, really specific. So it's almost able to go through into the genetic material, find the T cell receptor, take it out. And we're working on taking the endogenous T cell receptor out and putting our own T cell receptor in. That technology is coming along, but it's still a ways out for use in the clinic.
Okay, we've got a couple more questions. Do TCR-T therapies only target HLA peptides on the tumor cells? Do all tumors have these receptors?
So they do target the HLA peptides on, they can only target those HLA peptide complexes that they're usually on the tumor. However, if say another cell had them, it could target those as well. But that's why we put a lot of thought into what we target, because it should only be on the tumor. Not all tumors have these receptors. So they can come in a different flavor, or they can not have them very well at all. So that is another way that tumors can hide from our immune system, is they take away the HLA, so that making the tumors almost bald to the immune system. But there are therapies that you can administer that sort of help get those HLAs to the surface with those peptides in them. So there are ways that you can modulate them, and people are studying now combination therapies where they give that therapy that can help get those those HLAs to the surface, in addition to treating with T cells.
Another question, I can't remember if you said this already, but do patients receiving TCRs have any major side effects?
So with TCRTs, the side effects are a little bit more minimal. But with CAR Ts, you can actually have substantial, what they call cytokine release syndrome, or CRS. And so cytokines are signals in the immune system that tell the immune system to go or fire up. And so if you have too many of those at once, the immune system goes bonkers and starts attacking things that it shouldn't, including the central nervous system. So CAR Ts can have toxicity, mostly centered around too much inflammation and inflammation in the central nervous system, so the brain and the spine. Side effects can occur. Too much inflammation can occur with TCRTs. It's much less common because they are part of sort of that endogenous, that normal T cell and that normal interaction. So they're more susceptible to being turned off by those T regulatory cells.
Great. Why do you need to match the HLAs?
So you need to match the HLAs because the T cells, so they interact with the HLA peptide, and they touch, let's see, let's bring this up. So you can see that, maybe. So this interaction here. So you can see this T cell receptor interacts with the peptide in this pocket here, but it also interacts a little bit with the HLA on either side. And if you match those, so if you match a T cell receptor that has with an HLA that this T cell donor has, this interaction is fairly weak, and the interaction gets strong enough when this correct peptide is in the middle. However, if you have a T cell receptor from an unmatched donor, it actually interacts with the just the HLA, so just on these corners here, and that interaction is strong enough that it signals the T cell. So it doesn't matter what peptide's in the middle, but it reacts with the actual HLA presenting molecule. And if it's interacting with the actual HLA presenting molecule, that's on every cell in your body. And so if it's interacting with every cell in your body, you might be able to imagine how that's not particularly good. And that means that these T cells can see pretty much anything in your body and would target that as bad and turn you to a bag of mush, and that would not be good. Or if you have an organ transplant that's not entirely matched, that's what organ rejection is. And your own immune system is saying, that organ is not me, it needs to go. And so that's why we need to match those things, is so that our immune systems don't identify the foreign piece as being bad.
Great. A couple more now. How are TCRs infused into the patient? How long does that process take?
So that process takes shorter than you might imagine, maybe 30 minutes to an hour, and they're just infused how you would normally infuse maybe another IV fluid. It's done through the vein. So hang them up and they drip in.
And is it usually just like one treatment and that's it, or?
So it depends. So one thing that you need to get this therapy to work is those T cells need to hang around, right? And so sometimes the T cells, if they're not stimulated, they might just die and they won't hang around very long. And so sometimes it takes a number of different infusions of those T cells to get them to hang around in high enough levels to actually have an effect. So sometimes people get only one, sometimes people need multiple ones for sure.
And can a patient receive both CAR T cell therapy and TCR T therapies?
In theory, yeah, they could. They tend to be sort of mutually exclusive at this point, where people either working on TCR-T and CAR T. So no one has gotten double infusions yet, but that doesn't mean that it can't happen. They could get that one in theory.
Mm-hmm. Been having great questions from the students today, so good job, students.
Yeah.
I thought it was kind of interesting about, you know, like how the decision is made to pick a, you know, HLA that's more common in people, you know, of European ancestry, for example. And
Yeah.
you're kind of showing a little bit about, you know, how kind of there's a social dimension to some of the research and choices that we have to make. And,
Absolutely.
yeah. And hopefully there's like you say, that work progressing on other HLAs that can match folks in other communities.
Yeah, and that also comes a lot from donors too. So donors we get a lot of these T cells from healthy people who donate their blood to research, and we actually will identify these T cell receptors from healthy donors. So if we get
in terms of like the original research that you're doing, in order to understand.
Yeah, well, and to find these T cell receptors. So we engineer and we give them a very specific T cell receptor that can see cancer, but that T cell receptor actually came from a person.
Yeah,
so
that's interesting too. Yeah, so another reason why we need to diversify kind of the donor pool.
Yeah.
So I was wondering this too. Question: what's your favorite aspect of your research?
Oh, my favorite aspect of my research. Ooh. That's a hard one. I really love sort of the discovery aspect of it and sort of finding new things. So showing that processing is really important. People previously hadn't really done a lot of thought about that. So I think the discovery part is really, really fun. I like the techniques, but I think the sort of the thinking of new things is really probably one of my favorite parts of research, is coming up with creative ways of thinking. I used to always love science because there was a right answer and it was, you know, always defined, but research actually has a lot more of a sort of more English type skew to it, where you have to interpret your data and find what it means and then support it, and interpret your data and see whether it supports your ideas or not. So I think that's a really fun aspect of it as well.
By English, you mean like the kind of thing you would do if you were analyzing literature, or?
Yeah.
Kind of more the interpretation is important.
The interpretation is very important.
Yeah. And actually we see that a lot in our, especially our teacher programs, where if teachers haven't had experience with research, it's kind of astonishing, you know, how different school science can be from the research experience. Okay, two more questions. Were there any roadblocks you run into when working with TCR-T therapy? And if so, what's the biggest one you faced?
Many roadblocks. The biggest one I've faced, so when you generate these therapies, you put a lot of work into identifying a T cell receptor that can see and eliminate cancer, but then you have to do all of this, all of these safety checks, right? You have to make sure that if you give this T cell, it's not going to recognize something in the liver or the kidneys or the heart or the brain, so those essential organs. And so you do have to do a lot of safety testing, and a number of times you identify something that looks really good in the dish, it looks really great, and you start to do safety testing, and then you get to the end and it recognizes, you know, the lung, and then you have to go all the way back to the drawing board, because you can't put something in a patient that recognizes anything in the lung if it's trying to target your bone marrow cancer. So I think that's probably the biggest roadblock, is when you get to the some of the safety testing, which is it's a good roadblock to hit, but it does put you all the way back to the drawing board.
Great. Do CAR T cells also have to match HLAs, or do they recognize an antigen that is not presented by HLA? And also, follow up, as a grad student, do you get a lot of opportunities to give talks? You are such a great speaker.
Yes, lots of opportunities as a graduate student to give talks. Thank you. And CAR T cells actually don't have to be HLA matched because they recognize a protein on the surface of the cell. I think that's there. No, it's back here. No, forward. So CAR T cells, they recognize, oh, I don't have it. Yes. So CAR T cells recognize proteins on the surface of the cell. Now these proteins just exist on the surface. They aren't necessarily chopped up and presented on the HLA. So that's a huge asset to CAR T cells, is that you don't have to HLA match them. They do have an endogenous TCR, which again is usually virally selected. So that has potential to cause some of those issues, which they're working to get around with CRISPR, but for the most part, those get downregulated and don't cause as many problems. So you don't have to get a 100% HLA match because they recognize the surface protein. Good question.
Great. Couple related questions. How do you perform safety testing, and how long does the safety testing process take?
So it can take quite a while. It depends on sort of how many iterations you have to go through. So it can be anywhere from a few months to, over a year. And how you do this safety testing, you do it in a number of different ways to check yourself over and over again and make sure that it's not just a technical limitation of the assay that you're using to get that readout. So oftentimes you'll do sort of like those cold culture assays that I was describing to you. You'll use that, but you'll use a number of different tissue lines. So you'll use a cell line from the liver, from the lung, from the kidney, and so you'll use those. You can also use animal models to test some of the safety. Sometimes people will use mice or rats, sometimes non-human primates, although rarely, to make sure that the T cells are safe in those situations. But unfortunately, there's really only one way to trust true safety in a human model, and that's to use it on humans. So sometimes we do run into things there that we aren't able to test for in the lab space, and unfortunately realize that in early clinical trials. So that's why clinical trials will often start with just a few people, and often people who are very, very sick that have failed all of their therapies and have no other options.
And do you have any idea about how many people have been successfully treated by this method?
In total, I don't have a great idea. I know just in this clinical trial alone, we treated 24, and 12 did very, very well on that first arm. And I think about 2 or 3 might be still alive from the second arm. So that arm was much less successful. Mm-hmm. But there are many, many other groups doing this as well. So I couldn't tell you how many of those people did well or not, because sometimes they're published and sometimes they're not.
So we're still at the early days of this therapy, but it looks very promising.
Yes.
So it's really exciting. So we just want a reminder to everybody that our schedule is up online, and also the recordings from past presentations. So this recording will be up soon. And come back next week. We have Dr. Taran Gujral talking about how cancer cell social network shapes drug response, and so how they establish a social network that allows them to survive and escape therapy. So kind of another, cancer cells hiding related topic. And Dr. Gujral is an assistant professor in the human biology division. And so we're looking forward to that. And if you have a moment, feel free to give us some feedback about this presentation or any of your ideas for future Hutch Homes. I've dropped that into the chat. And you can feel free to unmute yourself or type in some words of support and thanks into the chat. And let's all give a round of applause to Miranda Lahman. Thanks so much for spending time with us today. And
Of course.
we learned a lot. Thank you.